Okay, welcome back. We're almost there. I know it's a war of attrition, I think, sometimes on these sort of meetings when you're sort of 4 days in. But last session of the day, we're going to go into some of the sort of biochemistry. So we've got 3 returning speakers who've all done some wonderful lectures so far. So we're going to start off with James Bennett again from the UK, who's going to go back into the hemolysis and AKI route. So James, please come and join us. Thank you very much.
Thanks, Richard. Hello again. Yeah, it's a pleasure to be back up here. And I've got a talk which is kind of following on from what I went through a couple of days ago, which was kind of the history of thoracoabdominal surgery and left heart bypass and what we could try and do to find some evidence on best practices. Whereas this is more, hopefully, a bit more futuristic. It's a bit more modern, and it's based around a study that we've been running for the last couple of years at Liverpool. And yeah, it's designed to be a pilot study. It's designed to tell us what we need to look into, rather than answer all questions about AKI in descending aortic surgery. But that's what it is, a hot topic of hemolysis and acute kidney injury following surgery on the descending aorta. So I am a fellow for Kidney Research UK. I'm also a perfusionist at Liverpool Heart and Chest Hospital, and I'm studying a PhD at the University of Liverpool. And I've got a couple of collaborations, one with the Centre for Proteome Research, which is based in Liverpool. And these very clever, generous people have done a lot of mass spec analysis for us. And also Biotech Support Group, which is actually a US-based company and has developed a novel polymer for the absorption of hemoglobin from plasma. But I have no financial declarations. So I'll just do a quick recap of the descending aorta and what the surgery involves, why patients sometimes develop acute kidney injury, and then I'll go into the imaginatively titled Hemo-AKI study and what the aims and methodology were before finally discussing what we've found so far. So, as we know, aneurysms of the descending thoracic and thoracoabdominal aorta can be life-threatening, but they're often asymptomatic. And when surgery is required, we as perfusionists can use several techniques to protect patients during this operation, including left heart bypass, deep hypothermic circulatory arrest, partial bypass, and selective renal perfusion. But despite all of this, we still see up to 50% of patients develop AKI after these operations. And of course, these are more extensive with, or more common, sorry, with more extensive aneurysms, but it's not uncommon with even extent 1 and DTA aneurysm repairs. So, as we know from several talks throughout the week, we've got ischemia reperfusion injury, we've got CKD, inflammation, and hemolysis, which all somehow contribute to this web of pathophysiology. And on the right there, you can see a typical plasma sample taken following surgery. And you know, as perfusionists, we look at that and we know it's the wrong color. It's a bit too pink. And it led to us really developing a hypothesis that we were just causing so much hemolysis during these operations that we were potentially contributing to acute kidney injury. Now on the left, you can see our historic technique for blood management during these operations. And that was a double cell saver system. So 2 reservoirs interconnected with a roller pump, and then a second roller pump, which can send the blood straight into the Belmont rapid transfuser and back to the patient's circulation. So some operations, we'd have 50 litres of continuous bleeding and reinfusion of unwashed blood into the patient's circulation. Now, it sounds crazy, but when the blood comes out very fast, you've got to get it back in very fast. And there's sometimes just no time for washing those cells when we need to just get them back into the circulation. Additionally, if we're just continuously washing litres and litres and litres of blood, we're going to remove all clotting factors in patients where bleeding is already a major risk. But when I started using this, I started to think, surely there's a better way of doing this. So we thought, let's design a study to investigate it. So a little bit about hemolysis. Obviously, the body has some mechanisms to try and take away some of the products of hemolysis. So hemoglobin, for instance, can be scavenged by haptoglobin, and then heme, which is a downstream metabolite of hemoglobin oxidation, can be scavenged by hemopexin. But of course, we didn't evolve to be on bypass. We evolved for the body to take away very low concentrations of hemoglobin and heme. And when they're exposed to crazy amounts of hemolysis, the body just can't keep up. And at that point, hemoglobin and heme pass through the kidney and they're filtered at the glomerulus. So we wanted to answer the question, is hemolysis a risk factor for AKI? So to do this, we had to ask ourselves a few things. One, what's the actual clinical impact of the AKI? How are patients faring? Do they have longer post-operative stays on intensive care? Are they more likely to pass away in hospital? Are they more likely to need hemodialysis? Does hemolysis contribute to AKI? Is point-of-care hemolysis testing something that we can do? And is it something that's useful to identify those patients who are most at risk? And finally, does hemolysis affect our kidney cells? So we wanted to have this model of taking it from the patient through to the organ and all the way down to the cell, where we can start to pick apart that pathophysiology and answer some of these questions. So how did I do that? Well, I broke this down into a hospital work package and a university work package. And when patients were consented to take part in the study, they'd come and they'd have their operations, but beforehand I'd take a plasma sample and a urine sample, and I'd do the same afterwards. In the bottom left there, you can see a device. It's called HemCheck, and it's a point-of-care device which uses 0.1 mils of whole blood, and it tells you the free hemoglobin concentration within 10 seconds. So it's a really quick way of identifying hemolysis. We'd then take those blood and urine samples to the university, and I'd run some assays on them. So I'd figure out, using lab standards, what the hemoglobin, heme, and free iron concentrations were, and the protein concentration. And in the middle there, you can see a patient's plasma that's been passed through a novel polymer called hemoglobin. And what that does is it adsorbs the free hemoglobin from the plasma. And you can see there's quite a nice separation in that tube, where we've got a nice red hemoglobin trapped at the top, a nice clear yellow plasma at the bottom. I could then take that plasma and we could look inside it, and we could see what proteins have been upregulated. And we could also see, if we apply those to cultured human kidney cells in the lab, how do they respond? Does hemolysis have an effect on those cells? Or is there something else in our AKI patients that might also be detrimental? And we do this through mass spectrometry at the Centre for Proteome Research. So ultimately, we've got a device that tells us how much hemoglobin. We've got a product that tells us what happens if we remove it. And finally, we can see what changes we have in patient plasma and in kidney cells. So we wanted to design something that was prospective, that was observational, and that was novel. And out of this whole process, we've got clinical data, we've got concentrations of proteins in the blood, we've got kidney cell proteins that we can identify. And we had 38 patients recruited to the study, of which 84% were supported with left heart bypass. We are the first study to use HemCheck for intravascular hemolysis detection. We're the first study to use hemoglobin, the polymer, for hemoglobin depletion in this way. And we've also got a unique application of what we call SIPTEX, which are immortalized human kidney cells that we use in the lab to replicate what's actually happening inside the kidney. So what did we find? Well, firstly, we looked at our patient characteristics, and you can see it's pretty ordinary. We had a decent creatinine concentration, we had standard body mass indexes, most patients were male, and we had 74% of patients who had thoracoabdominal repairs, and the rest had DTA. We had a 92-minute median cross-clamp time. About half of patients had custodial selective renal perfusion, and median kidney ischemia time was 19 minutes. So on paper, not too bad. Our free hemoglobin concentrations before surgery were very low, as you'd expect, 9 milligrams per deciliter, which is pretty standard. But then when we look at our outcomes, we see that exactly half of our patients developed KDIGO AKI. 29% of patients developed that to stage 2 or 3, so that's moderate or severe acute kidney injury. Almost a quarter of patients needed hemodialysis. Sadly, 2 patients developed paraplegia. Happily, no patients died in hospital after their operation. So we then thought, well, how do we possibly separate all of that? What are the most important results to focus on? And we needed to really define renal injury. And as we found earlier in the week, it's very difficult to do. And although we've got some standardized ways of categorizing acute kidney injury by severity, they're not perfect. And we wanted to find something that was somehow aligned to an international standard, but was also, it reflected what was actually happening in our patient cohort. So we used KDIGO, and essentially we took our stage 2 and 3 KDIGO patients, which is a doubling or more of serum creatinine concentration after surgery. And we found in this group, there was an increased need for hemodialysis, as you'd expect. 56 patients in this group needed it, and 0% in the no renal injury group. We had a 50% increase in critical care stay, so 5 to 10 days. And as I mentioned, happily, no deaths. So the first thing that we found that we were interested in was that patients with renal injury had a greater degree of hemolysis. So you can see on the box plot on the left, we've got patients in green who didn't have renal injury, and they had a median concentration of 97. So it's still a 10-fold increase in free hemoglobin concentration during the operation. But the patients who did develop renal injury had a much higher 17-fold increase, 167 milligrams per deciliter. Obviously, we've got a small sample size, and so it would be inappropriate to run a full logistic regression, but we did some univariate analyses, and we passed our significant outcomes into a Firth regression analysis, which is designed to take into account small sample sizes. And HemCheck point of care free hemoglobin was the only value that was statistically significant. Generally, although it's not a linear relationship, we could say roughly for every 1 milligram per deciliter increase above that median, the odds of developing renal injury increased by 1.5%. So how good is this test? Can we actually trust it? So we decided to find out, and we tried to compare the HemCheck values that we received from the machine with a lab standard. And you can see on the left there, we've got a pretty good correlation between the lab standard and between the point of care test. So we thought it's pretty accurate. On the right there, you can see a control chart, and we got one blood sample. We manually hemolyzed it through an insulin needle, and we repeatedly tested it with HemCheck. And obviously in a perfect world, every result will be exactly the same, but we're not in a perfect world. So a good quality indicator is that you have few of those points sit outside 2 standard deviations of the mean. Luckily, only 1 out of 30 fell outside that, and only just. So again, it's also quite repeatable. So we think it's a pretty good test to use. Now, how good's our polymer? What we were worried about when we used this polymer was that we would pass patient plasma through it, and it would actually somehow form some kind of toxic components that would end up killing our cells in the lab. So we needed to figure out whether it was biocompatible. And also, how effective is it? When we pass that plasma through, how much free hemoglobin, for instance, is actually removed? And you can see on the left, we've got a nice color change in the red plasma, which is taken straight after surgery, and the yellow plasma, which is then had that's been processed through the hemoglobin polymer. So just from a visual inspection, it looks pretty good. And then when you look at this graph, we can see that healthy plasma, and this is pooled plasma. So we got several patients and we pooled it all together. We can see a decrease from 180 milligrams per deciliter to 70. And in the AKI group, it went down from 290 to 85. And that can actually be improved by just simply adding more of hemoglobin to your plasma. So it was pretty good. I did some flow cytometry. So I took some of these kidney cells that we grow in the lab, and I applied either the unfiltered plasma or the filtered plasma to those cells for 24 hours. And then all you need to know from these graphs really is that each blue dot is a cell. If a cell sits in the green circle, it's alive. And if it's in the red circle, it's dead. And actually we can see that, and this could be pure coincidence, but the blood that had been processed with hemoglobin actually had more alive cells after 24 hours. So we know that the hemoglobin polymer isn't causing our cell death, which is great. What can we then discover from our patient plasma? So I took our plasma samples and went to the Center for Proteome Research, and they did liquid chromatography mass spectrometry on these. And what this does is it takes all of the blood proteins, it essentially heats them up, fires them down a vacuum, and then you can identify exactly what proteins are in that mixture based on the mass charge ratio. And that gave us 496 plasma proteins that we could identify. What we then needed to do was figure out, are there actually any big differences that we can recognize. Are we comparing apples and oranges here? The main thing we wanted to find out was whether there's a difference between post-op healthy plasma and post-op renal injury plasma. And you can see on this graph, we've got post-op healthy in yellow and post-op renal injury in green. And you can see that the ellipses that have been overlapped that are on the chart aren't overlapping, meaning that there is some difference in variability, which is a good start. So what we did next was we got all of these proteins that were identified and we put them on a volcano plot. And roughly what you can interpret is that if you follow the green arrow, these proteins are higher, they're more expressed in the renal injury plasma. And if you follow the yellow arrow, they're more expressed in the healthy plasma. And the higher they are, the more significant they are. And anything red is significant. So we've got a whole host of significantly upregulated proteins in our renal injury cohort. And these are interesting in themselves. For instance, if you look at the WIS protein, that's often something that is involved in sickle cell disease. So again, we've got some crossover potentially with red blood cells. But really, you can't take these proteins in isolation. You have to see, are there any groups of proteins that we know are involved in certain physiological mechanisms and pathways? And when we do that, and again, these results are only a week old. We haven't really delved into these into much depth yet, but I thought this was a really interesting start point. When you look at those proteins that are related to certain processes, we see things such as neutrophil-mediated immunity. And again, just opening that Pandora's box of immunity-related injury that we all kind of know about, but we haven't really unpicked yet. And I think it's a really good arrow. It's, our compass is kind of pointing towards this type of research now. But this is, in reality, what you get when you take all of your proteins and you put them into your database. You get very vague terms such as small molecule metabolic process. It's not very helpful, and it's really going to take us months to really delve into this. But I think we've got a good start. So, does hemolysis directly affect our kidney cells? So, our CypTECs, so our conditionally immortalized cells that essentially reflect the proximal tubule cells that we grow in the lab. They're a very good tool to use to investigate things like renal injury. When we take the patient plasma, we can batch it, we can filter it, and we've applied all kinds of conditions to our cells. We've then lysed those cells and we've taken out the inner contents for more mass spec analysis, and we don't have our results yet. We have sent them off, but they're still a few weeks away, so maybe next year. So findings to date, renal injury is associated with hemodialysis and prolonged recovery in our patients. Hemolysis is a significant predictor of renal injury. Point-of-care hemolysis testing seems to be useful, and there may be an immune component to renal injury. Obviously, there's limitations of the study. It's quite a small pilot study, which is what it was designed for. We knew we were never going to break any records for sample sizes. CypTECs may behave differently slightly to our normal kidney cells in the body. It's an in vitro application. We are a single center, and as always, there are plenty of confounding variables. So saying all of that, if anyone is interested in forming a larger study that somehow we can set a path and we can try and investigate things like immunity and how that might affect the kidney, please do get in touch. That's my LinkedIn, so if anyone wants to send a message to do that, please do. I'd just like to thank Kidney Research UK, who've been unbelievably supportive and very generous. Our amazing patients, of which 100% who were asked to take part in the study did so, and everyone who's part of our group and the perfusion team. Thanks.
Excellent presentation as before. So thank you, James. That's a great look at and an overview of everything. There's a lot of people making comments on the chat as well online, just saying superb, well done. I suppose the question would be, well, many questions, and I'll open up to the floor in a second. But can we, knowing that we're causing hemolysis is one thing, obviously, and then trying to do something about it. But when you mentioned last time that there is a sort of critical threshold for that. Can you remind me what that was?
So essentially we've found, so the median concentration for no renal injury was just under 100, and just over 100 was associated with renal injury. We did do some analysis and it worked out as just over 100, as to be, you were more likely to develop acute kidney injury.
And did you look, I know you said you had those patients that end up in stage 2 and stage 3. Was there a direct correlation between the amount of plasma hemoglobin and that stage? Or is it more multifactorial than
that? I'm sure it is multifactorial. Obviously, the downsides of having a small sample size is that you're really limited in terms of what you can find in terms of significance. The reason we chose the Firth regression model is that it accounts for small sample sizes. And actually, it does penalize you. So you're less likely to find significant factors, and it's more likely to compress those odds ratios as well. So something might look in a Firth regression analysis like it's not making much of an effect, but it is significant. And actually, the chances are, if you did have a bigger group, you might even see stronger outcomes.
Thank you. Great pilot study, I would say. Did you look at haptoglobin, like the capacity of getting rid of the free?
We haven't yet. It was something that we have planned to do, and we may well do. We've still got plasma in the biobank to do that. But we do know that haptoglobin typically is such a low level anyway, and our hemoglobin concentrations are so high that clearly they'll be saturated so early on that I don't think it's going to have much of an effect. But then there are other things we can look at as well. We can directly look at the cell, and we can look at things like ferritin, and we can see, our aim really with the SIPTEX is to see what's happening in the mitochondria. Is heme entering the cells? Is it then having an effect? How are our mitochondria responding to that? And so hopefully these are the kind of, but like you say, the more research you do, the more questions you have rather than answers sometimes, and you know it has opened a can of worms, but hopefully we can look into that.
Do you think that if you were to do follow-ups in the ICU, that the protein profile on your mass spec would change in some way?
I think so, because one of the great things about mass spec is that it tells you everything that's in there, but it doesn't tell you how it got there. It doesn't tell you whether the kidneys have expressed any proteins or there's some kind of cellular response. It could be that we're destroying or damaging cells. You know, it could be that the amount of haemolysis is related to the amount of proteins that are released from red cells themselves. So it's really difficult to pick that apart. And as we know, within 24 to 48 hours, that free hemoglobin will be removed by the kidneys, and I assume other things will also be removed or metabolized. And so, yeah, it's a good question. I think it'd be a nice study to do, maybe chart how that changes over the course of a few days after surgery.
I suppose following on from that to a certain extent is also that, you know, we were talking about the glycocalyx being damaged and fluids moving around, and obviously there's a whole host of, you know, sort of damage of, you know, the MPS molecules that will be potentially stored in the you know, in the interstitial fluid. And I suppose actually it would be interesting to look at the ITU course to see whether or not actually you had a sort of second hit on ITU as that then was brought back into the systemic circulation or even in the lymph, and just see how that actually affected it. I don't know if you've looked into that at all.
I've not looked into it. I thought Alex's presentation was really interesting earlier, and I took a few photos to see if there's any overlap between what we're doing. So yeah, it's something that would be really interesting to look into.
Any further questions? Yeah.
Sorry, everyone, I'll be quick. Did you look at the blood products that were sort of delivered during surgery, just thinking that obviously stored units, more fragile, more hemolysis? And did you manage to take that effect out?
Yes, so we've quantified the amount of red cell units and also the total volume of red cells delivered to the patients. And we found there was no significant relationship there on the univariate analysis. However, we have done an analysis on a larger cohort, a retrospective group, and the amount of red cell transfusions over the course of the patient's journey was significant. Of course, what we wanted to do was make sure we're taking post-operative blood samples as quickly as possible so we don't have interference with the mass spec from things like platelets or FFP, for instance. So we tried to capture that, and we did capture that before patients were transfused with those products. We typically use PRP and PPP, so some of the patients will have had their own proteins back, but no allergenic transfusions.
Perfect. Any further questions? No, in that case, thank you very much, James. It's brilliant. Thanks.